So these are human beings, and this is a fly, and they're separated by over 1.2 billion years of divergent evolutionary history. So you compute it by going back down one branch and then off on the other branch. And despite that, that's a long time even in evolutionary time, we're basically the same as flies. We share many genes with them. There are over 500 genes that cause disease in humans that have been identified with direct homologs in flies. And this is really going to be a story of yet another gene that was first identified in flies, then found in amphibians, and has been shown to turn up pretty much everywhere in living things, in fact, because it turns up in bacteria as well. And it seems to do the same job wherever you find it. So back in 1998, researchers who were investigating photosensitivity in the frog discovered a molecule in frog skin and later in frog retina that they called melanopsin. And when they sequenced the melanopsin gene, they discovered that it belonged to a class of photodetecting cells that included the photodetectors in the fly compound eye, the invertebrate compound eye. And remarkably, the melanopsin gene is more homologous to the invertebrate opsin genes than it is to the vertebrate opsin genes in the same eye. So rods and cones, the traditional photoreceptors in the vertebrate eye, also have opsins in them, but they're less related to this opsin gene in the same eye than they are to this gene in flies. And soon after discovering this protein in frogs and us are just kissing cousins pretty much, they found the same protein in humans, and they synthesized an antibody to it. And by synthesizing an antibody to it, to a protein, you can then go and stain primate retina and discover where in the retina this gene is expressed. And this is, as the person who gave me this slide describes rather poetically, this shows the expression of the gene. These fluorescent stripes are labeling the gene expression, as he put it, circling the foveal pit. So they don't enter into the fovea, the region of fine vision in the human retina, but they stain all around it, and then they also stain in the periphery. And just by looking at this, the people who did this staining work could see that these were not staining photoreceptive cells or traditional photoreceptive cells in the retina. They were clearly staining something called ganglion cells. And that was something of a surprise. So just to orient you to the human retina, for those of you who haven't seen this recently, this is a retina, and this is my laser pointer. I can only point on one side. No sides. Okay, so I'll tell you. So, and those of you who design optical systems would be horrified to learn, as many of you know, that light enters from the bottom of this system, and it travels all the way through these central parts, the wires of the retina, and ends up being detected at the top in these cone and rod cells, the things that are right at the top. And the rest of this mess here, which normally is transparent, which is why we can see it all, is wiring. It's wires and cells that multiplex and demultiplex the signals and form images and then send that resulting image off to the brain. And the ganglion cells are here. And so this was a surprise to find an opsin gene, a photosensitive molecule, being expressed in ganglion cells halfway down the wires of this photodetecting structure. And it'd be like finding out if you took apart a CCD camera that half of the imaging was happening in the wires that actually took the image from the back of the CCD rather than in where you thought it was happening. And just for reference, the giant sparse melanopsin cells are found at the top and bottom of this ganglion cell layer. And so what on earth is melanopsin doing in a ganglion cell? And the answer is it's detecting light. So you can knock out responses in the retina due to the rod and cone systems, and you can then record from these melanopsin-expressing cells. And sure enough, the red line at the bottom indicates when you turn a light on. And what you're seeing in the black lines here is spiking response in these giant melanopsin-expressing ganglion cells. And after you turn a light on for 10 seconds, these cells respond briskly. And notice two things. First of all, they take a little while to respond. So this is not a fast response like you get from a cone or a rod. This is a slow building response. And then it continues. After you turn the light off, this response continues for up to 30 seconds. So this cell is doing something very different to a rod or a cone, but nevertheless, it's detecting light. And you can show that it's genuinely detecting a different type of light to all the other photoreceptors in the eye by measuring its the spectral curve of its photodetection curve. And this is the melanopsin photodetection curve. These cells respond preferentially at about 483 nanometers, and all the other opsin genes in the eye peak at different locations. So this is a new class of photosensitive cell in the retina that was previously unknown. And since about 2002, it's been causing a lot of excitement. Two things that we know that it does, and these are probably the two primary things. First of all, it drives what's called the pupillary light response. So as I stare into this spotlight here, my pupils have shrunk down to tiny, tiny little dots. And that's caused by two things. It's caused by the rods and cones signaling that there's light, but also the melanopsin pathway has some role in this. At the top, you're seeing the pupil of a mouse. And the mouse is in the dark on the left, and then you take it into low light, and its pupil gets a little smaller. And then on the right, it's giving a talk at FiO, and it's in this very intense light. And in the wild-type mouse, its pupil shrinks down to nothing. At the bottom, you're seeing a mouse where both the rod and cone responses and the melanopsin genes have been knocked out, and you can see that there's no way that you can constrict that mouse's pupil. And if you only knock out the rod and cone genes, you find that it'll constrict, but then it'll come back. And this has solved a problem that's been known in the literature for over 100 years now, which is what's called the paradoxical light response of the pupil. And what happens is you take someone, you show them a very bright light, the pupil constricts, and then you take them out and you put them in the dark, and the pupil dilates again, and then it kind of bounces back. It constricts again after some time in the dark. And it's not clear why it should bounce back after some time in the dark and reconstrict. Well, now we know the answer. This pupillary constriction is being driven by two mechanisms. First of all, a rod and cone. Oops, sorry. Oh, it doesn't go back. Okay, I'm going to tell you. First of all, a rod and cone mechanism that drives an immediate constriction, a rapid constriction. And then secondly, a slower mechanism driven by the melanopsin pathway that causes a secondary constriction even if you sit in the dark for a while. And then the second and possibly even more important thing that melanopsin does is it entrains circadian rhythms in vertebrates, at least. And we can see this in mice, where the beautiful thing about mice is that you can do all sorts of unpleasant things to them and knock out different parts of their genetic pathways and their signal transduction pathways. And then there are now many mice running around the world where people have knocked out their melanopsin genes. So focus on the left for a minute. You can tell that mice are entrained to light because they wake up at night. This is the dark, I wish I had a pointer. The dark stripes in this diagram on the right represent dark times, and the black dots in there represent mice getting up and running around on their wheel. And this is how you tell that a mouse is active. All mice like to do, one of probably three things that mice like to do, is run on their wheel. And they run a lot. They run apparently about the equivalent of one human marathon every night in mouse feet, inches, whatever they run in. And in a wild-type mouse, they just run. You turn the light on, they go to bed, you turn the light off, they run, and it's very, very regular. If you turn the light off completely, they have their internal circadian rhythm. Circa means roughly, the rhythm that's roughly a day is actually not quite a day, and it starts to drift. So you can see in this dark period, about 2/3 of the way down on the right, if you turn the lights off completely, these mice still have this very regular rhythm, but they started to drift. And if you turn the light on, by the way, they don't know what to do, and they just become depressed and don't run on their wheels. On the left, you're seeing a knockout mouse, a melanopsin knockout mouse. And this mouse has relatively normal behavior for a start, and then you can trigger the destruction of all the melanopsin cells, so that you can actually knock out the melanopsin cells on the fly, as it were. And once you do this, you see that even though the light is coming on and off, the circadian rhythm is no longer entrained. The circadian rhythm starts to drift, just like a wild-type mouse is drifting in the dark. This circadian rhythm is drifting in the light. And it can be shown that melanopsin genes are essential for maintaining this strict circadian rhythm in mice. And notice also that this mouse doesn't get confused when the light is on all the time. It's as if it just doesn't understand that the light is coming on and off, and it has no circadian rhythm to set.